It's a pleasure to welcome you back. We're going to embark upon a second session. The topic here is human microbiota, atherosclerosis, and metabolic syndrome. As we discussed earlier this morning, there are a growing number of otherwise complex, already complex, clinical phenotypes, important diseases that now have very intriguing, at the least intriguing, links to activities of the microbiota. This is a great example. There could have been others that we could have run very interesting sessions on, including, for example, the products of the microbiota and their relationship to chronic kidney disease. Some of you know some of the emerging interesting observations there. But this is a topic that already has yielded some very interesting data. Stan Hazen is among the leaders and most interesting contributors to this particular topic, and so it's a great pleasure to introduce him as the moderator of this session. For those of you who don't know him, Stan Hazen is chair of the Department of Cellular and Molecular Medicine and section head of Preventive Cardiology and Rehabilitation at the Cleveland Clinic. Stan, again, has made major contributions in this area, and I know will lead a very interesting discussion. Stan Hazen.
Thank you very much, David. First, I'd like to introduce our panel. Immediately to my left is Dr. Arthur Reingold. Arthur is the Edward Ponhodt Distinguished Professor of Global Health and Infectious Disease and Professor of Epidemiology at the School of Public Health at the University of California in Berkeley. He holds concurrent academic appointments in the School of Medicine at UCSF, as well as serving as the head of the Division of Epidemiology at UC Berkeley. He has devoted over 30 years of his life towards the study of prevention of infectious diseases in the United States and various countries, such as in Africa, Latin America, and Asia, with a particular interest in vaccine-preventable diseases, including influenza, strep pneumonia, and Neisseria meningitidis infections, as well as other causes of pneumonia and meningitis. Dr. Reingold served on the World Health Organization's Strategic Advisory Group of experts, whose primary advisory committee on vaccines and immunizations, including as its deputy chair, and currently he serves on the advisory committee on immunization practices of the United States Department of Health and Human Services. As an elected member of the Institute of Medicine, he has served on multiple IOM committees, including most recently the Committee on Identifying and Prioritizing New Preventive Vaccines for Development. Dr. Reingold also has served as the president of the Society for Epidemiologic Research and is currently the president-elect of the American Epidemiological Society. And then Dr. Alan Addy, to the far left, is the Jack Gorski Professor of Biochemistry at the University of Wisconsin in Madison. He obtained his research training at the University of California in San Diego, studying lipoprotein and cholesterol metabolism. He's continued work in this area after joining the biochemistry faculty at the University of Wisconsin in Madison, and then in mid-career decided to study the genetics of diabetes using inbred mouse models. Dr. Addy's studies have employed genetics as an anchor point for integrating diverse phenotypes into causal network models. These diverse phenotypes range from physiologic traits to the many measurements delivered by omics technologies. He's currently working on use of genetic screens that will integrate microbiome phenotypes with traits related to obesity and diabetes. Thank you very much for joining us. Well, today I'd like to introduce the topic of the role of the microbiome in cardiovascular disease and in metabolic syndrome. As you heard, my background is both clinical in terms of preventive cardiology as well as basic science research in mechanisms of atherosclerosis. And one of the things as a clinician that I'm most struck with is we have many tools at our disposal for treating cardiovascular disease, but truthfully they're woefully inadequate. We have to move beyond the cholesterol-centric era, because we've figured out how to pound down cholesterol levels to exceedingly low levels, and yet still the remainder of events continue. This has been called the cardiovascular residual risk, and some have called it the forgotten and untreated individuals. Can I have an advance of the slide, please? There we go. So this slide illustrates some of the residual cardiovascular risk if one looks at the major multi-center statin trials in both primary and secondary prevention over the last 2 decades. And so starting with primary prevention studies, those are subjects without known heart disease, such as Forest or west of Scotland, all the way to the most current high-potency statin intervention trials, such as Prove-It, TNT, and Jupiter, where the achieved LDL cholesterol levels are shown as being exceedingly low, almost at the level of what they are when we're born, in the case of Jupiter trial, where the average LDL cholesterol is less than 60. And yet still, almost 50% of the events continue. So shown at the bottom in red are the percent of the residual events that continue to occur despite being on the high-potency statins. So while we know that cholesterol is mechanistically central to the development of disease, by the time people present in middle age, they have significant disease and significant events continue despite our current therapies. So one of the major areas that our group has been focusing on is trying to identify new pathways that are linked to cardiovascular disease. And this reminds me with what the topic of the overall session of this whole proceeding today, and that is that there's an entire universe within that we haven't addressed. And so when our studies began, and this was about a decade ago, I kept telling people, this is just like Men in Black 2. And the people in my lab kept saying, what are you talking about? And I said, well, this is a locker in Grand Central Station where Agent K opens it up, and there's a whole universe inside this locker. And that is what I think of as the gut microbiome. It is literally an untapped universe that we have yet to truly start to solve its intricacies of and how it contributes to chronic degenerative diseases, including cardiovascular disease. Now, the concept that microbes might contribute, gut microbes contribute to a complex metabolic phenotype, really began with studies that were put forth by Peter Turnbull and Jeff Gordon and colleagues at Washington University. And this slide illustrates kind of like the overall study that was performed. And if you look at the right-hand side, what they did, it's actually deceptively simple in its design. But showed, essentially, fulfilling one of Koch's postulates that you could transmit susceptibility for the development of obesity from a set of, by transmitting the microbes from donors into a recipient mouse. So two different inbred strains of mice that showed different obesity characteristics. The cecal microbes were harvested and transplanted by gavage into the recipient germ-free mice, maintained in micro-isolator bubbles, fed the same amount of food, and then over time their weights were monitored, and the mouse that actually received, or the mice that received the cecal microbes from the obese donors became obese, and the lean mice recipient donors, the recipients of the lean mice microbiome remained lean. Now, how did this happen? And so the Panel C shows the differences in weights that were observed, so it's quite a dramatic phenotype. If one looks at the left-hand panel, Panel A, if one looks at 2, 3, 4 carbon units of energy that are harvested in the cecal microbe content, the cecal slurry that's recovered from the intestines, you can see that there's enhanced 2 and 4 carbon units. So the bacteria were helping and aiding in the digestion of the food content within the intestines and making more small fatty acids available for digestion by the host. And shown in the middle panel, Panel B, is the results of bomb calorimetry experiments. What this is the feces was actually pyrolyzed, burned, and the amount of energy released quantified. And so the efficiency of energy harvest from the food was enhanced because of the microbes that were donated to the recipients from the obese mice. And so more energy was being extracted from the food, less energy was left behind in the feces. And this is at least one of the many mechanisms that we think this transmission of the susceptibility for obesity is achieved. Now, this was true not just with mice, but further studies by this and other groups have now confirmed the concept that the intestinal microbes contribute to the phenotype of obesity, including in humans. So you can take human fecal material and transplant it into mice and do the exact same type of experiment, and that has in fact actually been done. Now, what about other transplants? We heard from Dr. Relman earlier today about a rather crude fecal transplant experiment where very dramatic results have been seen by transplanting a healthy human fecal material into a subject or subjects who have recurrent Clostridium difficile infection, and with the results that there's superiority in terms of treatment efficacy with the fecal transplant compared to vancomycin. So it raises the possibility, could we have actually something of the effect of fecal transplants or some other targeted therapy that's based on intestinal microbiome that would be used to treat other cardiometabolic diseases. There are actually now feces in a capsule that are coming out and being treated in clinical trials. So this is not beyond the realm of possibility. And also, when we talk about metabolic diseases, there have already been studies, such as the one I show the reference for below, that actually have done fecal transplants, or duodenal infusions in this case, from subjects who are apparently healthy, lean individuals into subjects with metabolic syndrome, and were able to show improvement in insulin resistance and glycemic control in the recipients of the healthy donor feces transplant. So it is a possibility. Now, what I'm going to talk to you about a little bit is the connection between gut microbiome and cardiovascular disease, in case you haven't heard of this evolving story. What is shown on this slide is a summary of results of a variety of studies that have shown, as you heard, tantalizing connections between the gut microbiome and the development of cardiovascular disease. Now, we're all familiar with the concept that foods abundant in cholesterol and saturated fats are potentially a risk factor for the development of heart disease, such as the ones illustrated on this plate. But we also, when we eat these foods rich in cholesterol, ingest many other macronutrients. And so, for example, phosphatidylcholine, which is the major building block of cell membranes, or lecithin, is an abundant nutrient that goes hand in hand with cholesterol content. You can't have high triglyceride or cholesterol without it being bounded by a membrane in a tissue. And so almost every foodstuff that's high in cholesterol or high in saturated fat or triglycerides is also going to be high in lecithin. So when we ingest lecithin and another dietary nutrient, which has a very similar structure, and that's carnitine, they share this trimethylamine moiety that I circled in red. When these are ingested, the majority of it gets absorbed and digested the same way our medical textbooks describe, but some of it passes through the small bowel, is not absorbed, and makes it to the cecum and the large bowel, where we have bacteria that can then act on these foodstuffs and use it as a carbon fuel source. And what actually happens is the piece trimethylamine gets liberated. That is a gas at actual body temperature. It's what gives rotting fish the smell of rotting fish. It's a very objectionable odor. And yet we don't smell like rotting fish when we eat those foods, and that's because very efficiently through the portal circulation, the trimethylamine then is metabolized by a cluster of enzymes called flavin monooxygenases. These are enzymes that are like cytochrome P450s. They're involved in metabolizing especially biogenic amines. And what you end up with is a compound called trimethylamine N oxide. Now, I promise this is the only chemistry we're going to talk about today. But this compound, TMAO, is one that actually came up on untargeted metabolomic studies in human investigations for what compounds are associated with cardiovascular risks. So early on, in the beginning of the 2000s or so, when we began untargeted metabolomic studies, we were looking for new compounds and new potential pathways that might be linked to cardiovascular disease, and saw in both learning set and subsequent validation cohorts that TMAO levels in the plasma were tightly associated with risks of cardiovascular disease. Multiple subsequent studies have affirmed this now, including TMAO levels in thousands of patients has been shown to predict future risks of heart attack, stroke, and death, even independent of traditional risk factors in renal function. So then, that's just associative data. Why do I think it's mechanistically linked to the disease? Well, that's because hand in hand, additional studies were done where TMAO or the precursors, such as choline or carnitine, were actually fed to animals, and what was seen is they got accelerated atherosclerosis. So, for example, in mouse models of atherosclerosis, if you feed with choline or carnitine, you get accelerated atherosclerosis, but only in the presence of intact gut flora. If you don't have intact the microbes to make the TMA and make the TMAO, then the diet-induced enhanced atherosclerosis didn't occur. Alternatively, if you add the TMAO directly to the diet and bypass the microbes, you saw enhanced atherosclerotic plaque development. Now, mechanistically, how this occurred is quite complicated and not fully elucidated, but what is clear is that TMAO is a biologically active compound that has an obligatory role for gut microbes to be made in both humans as well as in animals, and that it will influence cholesterol and sterol metabolism in multiple different compartments. So, for example, in the artery wall, TMAO influences macrophage phenotype and inflammatory gene expression. In the liver, it's a very potent regulator of bile acid synthesis and composition, and transporters of bile acids and cholesterol metabolism. And in the intestines, it's also impacting both bile acid and cholesterol and sterol metabolism. So you can think of this compound as like the rheostat on a light switch. You need electricity to turn the light bulb on, just like you need cholesterol to have atherosclerosis. But if the rheostat is turned up, if you have a high TMAO, you have greater propensity for deposition of cholesterol in the artery wall. And when the TMAO level is down, you have less propensity for development of atherosclerotic plaque in cardiovascular events. So lastly, I'd just like to say that this pathway is really an interplay between not just the microbes, but the host as well. So it's a meta-organismal pathway. It involves both multiple microbial enzymes as well as the host hepatic enzymes that play a role in the overall development of the metabolite. So a couple of final take-home messages to leave as discussion points before we open up for the other panelists, and then the question and answer. First is that the gut microbiota appear to be a participant in cardiovascular disease, so therefore perhaps they can become a therapeutic target in the treatment of cardiovascular disease. And I'll talk a little bit about possibilities of how that might occur in just a moment. In addition, some additional take-home messages to discuss or think about are we really need to begin thinking about ourselves as walking communities. And when we talk about diseases, it's the composite of the metabolism between not only what we eat and what the microbes, the filter of how it's perceived by us, the host, through the microbiome. And so our biggest environmental exposure is what we eat. Technically, food is a foreign object that we ingest at the kilogram level. And if we have different filters, we're experiencing that outside, that environmental exposure differently between us because of the difference in the intestinal microbiome filter. As I mentioned, we also need to start really thinking about our metabolism and disease more globally. This is true not just for cardiovascular disease, but obesity, insulin resistance. Many of the chronic metabolic diseases are now showing links to intestinal microbe-dependent pathways. And then also, so what is the definition of a hormone? It's a biologically active compound that's made at one site and through diffusion or in the circulation is delivered to a distant site. If you follow that strict definition, the microbiome can really be considered as our largest endocrine organ. And many of the products that it makes are biologically active hormones. It's a plastic endocrine organ because what it makes depends upon what you feed it, and not both acutely and chronically in terms of the long-term dietary habits. But nonetheless, it's an endocrine organ, and that brings us to the final thing, is as soon as we can understand what the chemical signaling is, the braille, if you will, that the microbial symphony is playing, and what these participants that are being generated are in the pathways that lead to chronic diseases or participate in health as well, this can actually be exploited at the drug level. And so I would like to suggest that in our futures, we will begin to think of statins as our Homo sapien enzyme inhibitor to block cholesterol, but we will also have, perhaps in our medicine cabinets, not antibiotics, which are lethal drugs, but instead compounds that are non-lethal to the microbes and simply modify what on the platter or the plate of what's available for them to eat, they're able to eat. And so will therefore help prune the compounds that they generate and influence overall diseases such as cardiovascular disease, obesity, or diabetes. And with that, finally, what I'd like to suggest is that there are a whole large number of potential approaches one can take towards drugging, if you will, the microbiome. Now, one of the drugs, as I said, food is a foreign object, diet is a huge selective pressure that influences the microbial composition. And so clearly by chronic dietary exposure, you can change microbial composition and function, as well as by acute changes. I mean, if you don't give the dietary nutrient, the product doesn't get made. But in addition, there are other approaches towards manipulating the pathway, and which will come out further in the discussions. With this, I'll switch to Dr. Reingold.